Hello, my name is Emma Morris, and for my bioinformatics project, I chose to study the association and linkage of ABCA7 to Alzheimer's disease. So, the primary purpose of this project was to determine whether or not the gene ABCA7 is essential. So, an essential gene is one that is conserved with a slower rate of evolution, and a non essential gene is one that is non conserved with a faster evolutionary rate. So this project allowed us to apply an evolutionary perspective to whether or not variation between SNP and phenotype differences are likely to be important or not. The method, so I started with installing Eugene on my computer. Eugene is a computer software that is used specifically for bioinformatics. And then after this, the first step was to select a phenotype of interest as well as a genotype of interest, and I got both of these from the Genome-Wide Association Studies database. After I selected my genotype of interest, I identified the accession number for the human protein product, which I got from Wikipedia, and then I conducted my BLAST search. The sequences, I then imported them into the Eugene software, which is used to align the sequences so that the homologous comparisons can be used to construct the gene trees. Regarding the gene trees, there were three that were supposed to be made, but for me, only the neighbor joining tree ended up working. So I ended up with just one out of the three gene trees. And then following this, the next step was to calculate the pairwise genetic distance as well as the linear regression. And the slope from the regression is the estimate of the molecular clock for my gene as well. So for the phenotype of interest, I chose to do Alzheimer's disease. I chose this because I do have a personal connection to it, and I've honestly just always thought that memory loss was super interesting and an interesting concept to learn about. So, what is Alzheimer's? It's a type of dementia that affects memory, thinking, and behavior. Symptoms of Alzheimer's eventually grow severe enough to interfere with daily tasks, and it's a progressive disease. So, this means that symptoms get worse over time, and individuals who have it usually only live about 4 to 8 years after the diagnosis. So, getting into the molecular part of Alzheimer's, there are two abnormal structures called plaques and tangles, and they're thought to be the main causes in the damaging and killing of nerve cells. So, plaques are deposits of a protein fragment called beta amyloid, and it builds up in the spaces between nerve cells. And then tangles are twisted fibers of another protein called tau that build up inside of cells. So everybody across the board has plaques and tangles, but it's been shown that people with Alzheimer's have developed many more of them, specifically in areas affecting memory. So this is thought to possibly block communication between nerve cells. Next is the SNP results. So my SNP number was RS37522246. And according to a study by PubMed in European and African American populations, indirect ABCA7 genome-wide association studies are explained by expansion of an ABCA7 variable number tandem repeat, as well as a common premature termination codon variant. Next, my selected gene obviously is ABCA7. It has 47 exons and it's found on the 19th chromosome. This here is its protein accession number, and its family is the ATP binding cassette subfamily A member 7. Next up is the alignment. I aligned the sequences through Eugene, which allowed the system to match up the nucleotides. And then once aligned, the algorithm was able to detect any relationships between the sequences. And so from this, I was able to see if they are ortholog or paralog sequences. And the difference between the two is that paralog are similar because of convergent evolution, and ortholog has a